Good afternoon, everyone. I'm Dr. Rena Reyes, and I would like to welcome you to the Spinal Cord Injury Forum on this beautiful June day here in Seattle. This is a University of Washington program that is brought to you by the Northwest Regional Spinal Cord Injury System and made possible by grant support from the National Institute on Disability and Rehabilitation Research. Today, our keynote speaker is Dr. Chet Moritz, who received his undergraduate degree from the University of Washington in 1998. He then received his PhD from University of California, Berkeley, where he studied biomechanics of human movement. He completed a postdoctorate at the University of Colorado, where he investigated neural control of precision hand movement. And because that wasn't enough, apparently, he completed a second postdoc at University of Washington, where he began his interest in brain-computer interfaces and neuroprosthetic technology to treat paralysis. He is now an assistant professor in the Department of Rehabilitation Medicine and Physiology and Biophysics. He was recently named an Allen Distinguished Investigator and serves as the Deputy Director for the Center for Sensory Motor Neural Engineering. In his efforts, he collaborates closely with engineers, neuroscientists, and clinicians to develop next-generation technologies to treat spinal cord injury and other nervous system disorders. His work has been published in the journal Nature and actually featured on national television news. Today, he will be describing the state of the art in the field of neural engineering for the treatment of spinal cord injury, using examples from his own work as well as those of his colleagues around the country. Please help welcome Dr. Chet Moritz.
It's a pleasure to be here today to see some old friends and familiar faces. The overview of my talk is this idea of interfacing the nervous system with electronic devices to improve function after paralysis or paresis due to spinal cord injury. And I'll show you several examples both from my own work and some very recent clinical trials that are ongoing around the country that illustrate the potential benefits of this type of treatment. So the overall goal of this work is to restore limb function, although we'll talk about some exciting anecdotal findings for bladder, bowel, and autonomic dysreflexia as we go through. And you can really divide this goal into two main outcomes. And the first outcome I'll talk about is this idea of enhancing movements or enabling movements through the use of electrical stimulation. And in some cases, in some of our animal models, we've actually seen these improvements persist beyond the period of stimulation, which I think is quite exciting. The second part of the talk, I'll deal more with what I'll call direct reanimation. This would be the case perhaps in a complete spinal cord injury. We'll use the example of tetraplegia or quadriplegia, and our goal there is to reanimate the hand and arm or restore function to the hand and arm through computer devices. So an example is shown in this sketch here, and by the end of the talk today I'll show you our initial example in animal models that this type of approach is feasible. Here we're recording the activity from within the brain, recording the intention to move or the desire to move, translating that into stimulation of the spinal cord below the level of the injury. And this diagram, I think, serves nicely to illustrate the main components of my talk today. We'll begin with a study, an ongoing clinical trial by Susan Harkema at the University of Louisville in Kentucky, where she's stimulating in the epidural space over the surface of the lumbar spinal cord to enable movements of the lower extremity. And we'll show some videos, and there's some very recent results published now on a total of 4 subjects who are either Asia A or Asia B, able to regain some movement of their lower extremities when the stimulator is operational. Then I'll tell you about some work in my own lab where we are stimulating within the spinal cord rather than on the surface of the spinal cord. And we have dual goals with this intraspinal microstimulation. One goal is similar to Susie's, to enable movements when, in our case, the animal subject is attempting to make movements. And I'll tell you about, I think, some fairly exciting findings that this recovery, this enabling of movements, persists well beyond the period of stimulation, so there's some therapeutic effect. Our second goal with intraspinal stimulation is for reanimation, or for directly causing movements of the limb. And in the final example, I'll show you perhaps causing movements of the limb that are directed by the ongoing activity of the brain. So I'll summarize some ongoing clinical trials in what I'm going to call brain-controlled interfaces or brain-computer interfaces. We've really moved beyond just using these devices to control computers. We now have clinical trials where users are controlling robotic arms, There's a clinical trial that's just begun where users are controlling stimulation of their own paralyzed muscles to restore movements of the digits. And so we'll briefly summarize the work there as well as some of the animal studies that led up to this. So let's begin here first with the epidural stimulation study by Susan Harkema. Epidural stimulation, as many of you I'm sure know, is FDA approved for the treatment of chronic pain. So there are devices out there which look a lot like a pacemaker, actually. The can with the battery and the electronics sits in the abdomen. There's a wire that runs up to the spinal column, and the electrode is inserted through a small hole drilled in the lamina, the vertebral lamina, and that electrode typically lies over either the dorsal roots or the dorsal surface of the spinal cord to provide some relief from chronic pain. Now, for about the last 10 years, individuals have been using similar techniques in animal models to stimulate the surface of the spinal cord and enable locomotion or enable stepping, especially when that stimulation is coupled with the movement of a treadmill belt, weight-bearing support, and at times enhanced by treatments such as quipazine, which is a serotonin agonist or 5-HT agonist. And so all of these animal studies paved the way for the first human trial that Susan Harkema launched several years ago in Kentucky. And the first subject in her trial, his name was Rob Summers, and he was injured about the age of 20, almost 21, in a tragic accident, a hit-and-run car accident. He was standing by the side of the road, and a car drove by and hit him and then drove off. He ended up with a subluxation injury at the C7 to T1 junction that's shown here on the MRI, and he was classified clinically as Asia B. No motor control below the level of the lesion, and abnormal but some sensation below the level of lesion. So Rob participated in very intensive physical therapy for several years before enrolling in this study. And he did improve function to some extent, but still had no motor function in his trunk or his legs, and also flaccid anal sphincter and no voluntary control of his bladder when he enrolled in the study. The study involved placing an epidural pain stimulator, but in a different location and with a different pattern of electrodes. This, instead of being a single site or a small array, this was a large array of electrodes spanning the first lumbar to the first sacral spinal segment, so L1 to S1. And stimulation, instead of being tuned to alleviate pain, was tuned to excite the spinal cord or enhance the activity of the circuits below the injury. And so he would participate in intensive therapy for 40 to 120 minutes while the stimulator was running in what I'm going to call tonically. So it was stimulating a frequency of 5 to 40 times a second, but running the entire time, not being pulsed to try to evoke movements. And Rob participated in these studies for about 7 months. And then one day they noticed that he was actually able to control his legs when the stimulator was running. And so I want to show you several videos that illustrate that exciting effect. This first video, you should hear on the audio the narration of the experimenter asking him to produce different movements. But in this case, the stimulator is turned off, and so you won't observe any movements in this first video.
Left toe up, left toe down, left ankle up, left ankle down, up, left leg down, toe up, left toe down, left ankle up, left ankle down.
Okay, so no movements observed there. This is an experiment that immediately followed. Now the stimulator is turned on, but just running the entire time. Okay, and observe what Rob's able to do with his legs.
Left toe up, left toe down, left leg up, left leg down, left leg up, left leg down, left ankle up, left ankle down, left ankle up, left ankle down, right, left toe up, left toe down.
A fairly exciting and impressive example. Susie's gone on to repeat this now with 3 other subjects. Rob is shown here in the first column. He was Asia B upon entering the study. They enrolled 2 individuals who were Asia A and another who is Asia B. Now in Rob's case, they didn't actually ask him to produce movements until after 7 months of training. The goal of the study was not actually to improve movement. The goal was to study conduction within the spinal cord, if it could be enhanced with stimulation. It was actually a surprise to everyone, including the experimenter, when one day while the stimulator was running, Rob said, "Hey, Susie, I can move my toe." Right? And she was quite surprised and asked him to repeat it. There were several excited exclamations exchanged between the two, and then they discovered perhaps a new intervention. So in these second 3 subjects, even after 2 weeks of physical therapy combined with stimulation, they were able to produce movements. There's the upside is that Rob continues in the study. All these subjects continue in the study. Even when the stimulator is off, most of them have improved bladder, improved bowel, improved AD, less incidence of autonomic dysreflexia. They are not, however, able to move their limbs, other than perhaps the faintest toe twitch in the case of Rob, when the stimulator is not continuing to run. So to try to address that limitation, see if I can get rid of this bar at the bottom there. To try to address that limitation in our lab, we are exploring intraspinal microstimulation with the idea that placing hair-like wires within the spinal cord may allow us to activate spinal circuits more selectively and may result in a better therapeutic outcome. And so this slide here summarizes the difference between the 2 studies. So at the top, what I just told you, with rare exception, epidural stimulation, this is what the grid of electrodes looks like that was used in Susie's study. It enables movement while the stimulator is running, not after the stimulator stops. Intraspinal stimulation are these tiny wires which actually insert within the gray matter of the spinal cord, thereby activating local spinal circuits. And so in our lab, we asked the question whether this intraspinal stimulation could lead to improvements that persisted beyond the injury. And to ask this question, we used our animal model, the rat, and we used a model of contusion injury, lateralized cervical contusion injury at level C4. Before injury, the rats were trained to produce what we call a precision 4-limb reaching task. They had to reach out, grab a food pellet, bring it back, and eat it. That involves grasping, supination, cortically controlled movements. The rats were then subjected to a lateralized C4 contusion injury, shown here in the diagram, before they were implanted with stimulating electrodes below the level of the injury. Beginning 4 weeks after injury, which would be considered a subchronic or semichronic time point in these animals, we began stimulating therapeutically 7 hours a day for a total of 12 weeks. And then we would measure the animal's ability to perform this task with and without the stimulator active to look for sustained effects. And what we observed was that animals indeed were able to perform the task much better after they had undergone this stimulation treatment. So this first animal was never stimulated in the therapeutic sense for the duration of the 12 weeks. And so this provides a baseline level of deficit. The goal is to grab this food pellet here. The animal is kept interested by the experimenter holding another food pellet. And you can see that the injured limb, unable to break the plane of the arena and acquire this pellet. By contrast, a stimulated animal, after 12 weeks of therapy, but with the stimulator disconnected, is able to easily reach and recover the pellet. Not fully recovered, she stumbles on the way back. It's difficult for her to weight bear on that limb, as you'll notice, but much improved compared to the animal who is not stimulated. And that held true across our group of 11 animals in each of the groups. The stimulated group shown here on the top, a modest but significant improvement compared to the unstimulated group shown at the bottom in blue. And in one animal's case, we had the rare opportunity to observe how long this improvement would take place. This animal actually had an implant failure after 5.5 weeks of stimulation, so at the end of the black bar here. And while this animal recovered to nearly pre-injury levels before the implant failure, there was a subsequent loss likely due to failure of the wires in the spinal cord, and then a very nice sustained recovery well above the level you would expect from an untreated animal, which would be down here near 5 or 10%. And the mechanism appears to be, in these studies, appears to be improvement in tone or spasticity, especially flexor tone. So these animals exhibit the flexed posture of the digits, flexed posture of the upper extremity, as is common in human spinal cord injury as well. And serendipitously, stimulation within the spinal cord preferentially activates movements that counter that flexed posture. And so here we see the most common movement evoked is extension of the elbow, which helps in that reaching task. The second most common, which actually becomes more prevalent over the weeks of treatment, is extension of the digits or opening of the paw. And so these spinal stimulation effects map directly on a more quantified version of the reaching task. So if we look at the animal's ability to aim, advance the limb, extend the digits, and pronate the paw, they're all significantly improved and quite related to the spinal stimulation effects. And you can see in the stills here, a stimulated animal able to open the paw compared to an unstimulated animal where the elbow is flexed and the digits are also flexed. So we think our mechanism here is likely acting via improving tone, possibly via the serotonergic system. So I just want to summarize the first part of the talk here by contrasting what we believe the mechanism of epidural stimulation is from the first study with Susie and Rob, with the mechanisms in our later studies. We think that epidural stimulation, at least for movement enhancement, works by bringing the spinal circuits closer to threshold so that remaining descending drive from the brain, remaining cortical input is able to cause those circuits to move over threshold and cause a movement. So obviously individuals in this case, they might be clinically complete Asia A, but they're likely not anatomically complete. There's some level of brain control still over the spinal cord. By contrast, in our animal experiments with intraspinal stimulation, these benefits seem to persist, and we think that we're likely re-regulating the excitability of the spinal cord, perhaps through serotonergic mechanisms. 5-HT is another term for serotonin, to improve tone and spasticity. And anecdotally, we even see some of the earliest stimulation sessions, we immediately see tone relax. When the stimulator is shut off, it comes back. But over weeks, the more time that the tone is absent, the time becomes longer, and eventually the stimulation is not needed to relax that tone. It's just interesting to note that studies have shown in human subjects and animal models that functional electrical stimulation, or stimulation of the peripheral muscles or nerves, as is done for FES cycling and other types of therapy, have also noted improvements in tone and spasticity. So it may not be unique to stimulating within the spinal cord, and it also may be accessible in the time that it takes these ideas to get to clinical trial. So I'd like to move on now to the second portion of the talk here, where we're going to discuss the idea of brain-controlled interfaces. We're going to change gears a little bit and now think about someone with complete paralysis, or Asia A, and in my case we're going to think about someone with tetraplegia or quadriplegia. And so our goal is to restore function to the hand and arm, either directly by stimulating the paralyzed muscles or via a robotic assistive device. And so the field of brain-controlled interfaces, otherwise known as brain-computer interfaces, has advanced quite a long way, even in the last 10 to 15 years, from initial demonstrations in animal subjects all the way through ongoing clinical trials now at multiple sites, including Brown was the first site, Case Western Reserve, Stanford, Utah, Ohio State. There's many sites now ramping up. So it's something that's becoming more accessible. And the idea with the brain-computer interface is that you record the intention to move from the brain, typically from primary motor cortex. There's a nice map, a somatotopic map of motor cortex shown here, where the hand and arm area has a very large representation because we have such dexterous control over our digits, at least before injury. And so electrodes placed in or near that part of the brain can provide information about the goal to move. There's several different ways that you can extract that information. The least invasive, but the one with least information, is scalp-based electroencephalography, or EEG. And here you see a cap of 120 electrodes worn on the surface of the skin, and they can pick up the brain waves as they propagate through the skull and through the scalp. But the analogy I like to use for EEG is it's like listening to this talk, to this lecture, from outside of the building. You can tell that the lights are on, you can tell that something's happening, but you don't get very detailed information about the type of movement the user is planning. You might get information about move or don't move, or which part of the body perhaps they're attempting to move. But to get that kind of detailed information, you need to take a step closer to the brain. And this idea of electrocorticography, or ECOG, is clinically used to diagnose individuals with epilepsy, to figure out epilepsy that's intractable to medication, to figure out which part of the brain needs to be resected. So individuals are implanted with these grids for a period of several weeks and can then participate in brain-computer interface studies. And this is like listening to this lecture from outside the door. So now you can hear my voice modulating, you have some idea what's going on, but you're still perhaps not catching every word, you're not seeing the details on the slide. And to see those details, you need to come into the room, sit in the front row, And here is an example of doing just that in the brain, and that's placing tiny hair-like wires into the brain to record from the single neurons that are actually coding this movement, controlling this movement. And these are the studies that I'm going to describe from our lab, as well as the studies that are ongoing in clinical trial. So I think we'll skip over some of the slides in your handout there just so we can get to the exciting results. And so we'll look at the first human study of this implanted array. And this is this idea of what we call the Utah array, designed at the University of Utah. It's 4 millimeters on a side. So here it is on the head of a penny, absolutely tiny. The long wires you see just tether it to a connector. This will all eventually be wireless. There's people working on that. And this is the array implanted in hand area of motor cortex. That's this enlargement here, this knob we call it. And so this was the first subject implanted with the Utah array. He's an Asia A, tetraplegic, from a stab wound, I believe, at least 4 years prior to this study. And using the activity recorded on these electrodes, he's able to control the movements of a computer cursor in this video that I'll show you next. So he controls the blue dot on the screen. And the game he plays is to try to move the blue dot, the cursor, over targets that make happy noises, like money bags that go cha-ching, and animals that bark. And he's supposed to avoid the evil targets, the ones that make evil laughs. And that will become fairly apparent, I think, as you see the video.
Good. That was a great one. Good.
Damn it.
And so hopefully you can appreciate that it's useful control, perhaps not nearly as perfect as you might do with an able-bodied user using a computer mouse. And if you were listening very closely, you could hear that he was also very motivated, a little bit frustrated when he made 2 mistakes in a row. This was an example of using about 30 neurons, all decoded. Each neuron has a different preferred direction in imagined movement space. So he imagined making movements, they could tell which neurons coded movement in which direction. So he could control that computer cursor. This same group in their next subject was able to demonstrate control of a robotic arm, and that's shown here. This woman actually has a brainstem stroke rather than a spinal cord injury, but her task is to pick up this bottle of coffee with this robotic arm, and she's controlling the movements of that arm with the activity recorded from her brain, same type of electrode. There's a few tricks here. They're moving the robotic arm in the plane of the table until she issues a command to grasp, and she lifts it, and again it's in the plane of the table there until she issues a command to tip and drink from the cup. She's concentrating very hard and accomplishing her goal, although as you can see it's slow compared to typical movement. This is her view down here in the bottom corner. When she gets close to her mouth, she's able to tip the cup and drink from the straw during that time. So obviously a very gratifying moment for both her and for the experimenters to see that this device could provide some sort of assistive function. And then she proceeds to put it back on the table. And I will skip over this part because it looks a lot like when she picks it up. What I want to highlight, though, from the stills in this video is that this particular implant was 5 years old at the time that this video was shot. And one of the current challenges in the field is recording from electrodes in the brain for long periods of time. We know that these electrodes degrade over time. The brain throws up a protective scar, a lot like the scar around a spinal cord injury. And so the recordings that were left on this array were perhaps only 3 particularly strong neurons. And so she's essentially controlling this robotic arm with about 3 different neurons in her brain, which I think is very impressive if you think of how flexible the brain is and how well it can learn to use these devices. Just about a year later, this was just last year, another group implanted now 2 Utah arrays at each of the black diamonds here in a woman with spinocerebellar degeneration. So she was otherwise paralyzed from the neck down, and they performed an MRI to look at which parts of her brain were active during imagining movements of her hand, and the yellow area is the hand movements here. They implanted these dual electrodes, they trained her in a virtual reality environment, and then they asked her to control a real robotic arm with her brain activity alone, and that's what's shown in this video. So there's no audio here, but she's controlling 7 degrees of freedom, so 3 degrees of freedom in space, 3 degrees of freedom of rotation at the wrist, and 1 degree of freedom of grasp. And again, control is nowhere near perfect, but she's performing now some of the tasks from tests of function. So a cup stacking task, if you're familiar with that, you'll see a box and blocks task next, or modified box and blocks. And then it just takes her several tries sometimes to grasp the object, but imagine the challenge now. She's using a part of her brain to completely control the movement of this robot. And here she re-grasps the cup when she's not happy with the original grasp, and she pours this pink cube of paper, which is a simulated liquid. Her goal is to pour that into the mixing cup here, and she gets very, very close to doing so. And if you watch closely, unfortunately, the paper cube falls just behind the cup, but it looks, from the video angle, it almost looks like she succeeded. So let's call it a success. So I think these are all extremely impressive examples, extremely inspiring examples. But if you survey individuals with tetraplegia, or quadriplegia as it was called until fairly recently, as Kim Anderson did in 2004, and you ask them what the highest treatment priority is, it looks quite different than paras, right? Paraplegic treatment priorities are bladder, bowel, sexual function, and then perhaps walking. For a quadriplegic or tetraplegic, regaining use of the hands and arms is by far the #1 priority, and about 5-fold higher than the other items included in this survey. And so the goal in our lab about 6 years ago now, I guess, was to try to merge the technologies that you just saw, recording from the brain, with existing technologies of functional electrical stimulation or artificial stimulation delivered to the peripheral nerves. And at the time, there were commercially available systems like Hunter Peckham's Freehand that could help individuals with a C6 injury to restore grasp by shrugging the opposite shoulder, for example, and about 40 years worth of research in FES for both locomotion and for reach and grasp. And so our goal in our animal model was to try to combine these two fields for the first time. And we chose to do this in a monkey model because the monkeys were smart enough to learn the task and perform the task. We, of course, didn't want to induce a spinal cord injury in our monkeys, so we developed a reversible nerve block technique, similar to the kind you'd have if you had a hand surgery, where we can inject lidocaine into the nerves as they leave the shoulder, and for about 2 to 3 hours, reversibly paralyze the arm and hand. Then we asked monkeys to play a video game, which I'll show you an animation of in a moment, but instead of being able to play the video game by moving a joystick with their wrist, as they typically do, they had to use the activity of neurons in their brain to control stimulation delivered to these otherwise paralyzed muscles. So they had to reanimate the wrist, in our case, in flexion extension, using brain activity alone. And that's what's summarized here in this animation. Each of the experiments began by the monkeys directly controlling the activity of the neurons as viewed by the movement of a cursor on a screen. And that's similar to the original brain-computer interface studies that we described, moving the blue dot. Here we have a white square instead of a blue dot. Once the monkeys had demonstrated control, we used what we call the neurochip, which is a small cell phone-sized stimulator, to convert the activity recorded from the brain into real-time stimulation of the paralyzed muscle. So now the monkey changes the activity of the brain cells to change the amount of stimulation delivered to his wrist and move his wrist or produce torque about his wrist to acquire targets on the screen. This is an example of the level of control achieved by the monkeys if we plot across here the time, total of 12 minutes shown here. And the monkey's task is to acquire one of these 5 different targets by flexing or extending the wrist. Center target shown in gray, and the monkey could rapidly move back and forth as these targets were randomly presented. And modulating the neural activity is shown by the histograms here, high firing rate of a cell, low firing rate of a cell. And the key is that every 10 minutes we would shut the stimulator off for about 30 seconds, and we would ask the monkey to try to acquire these targets. And you can see by the thin blue and thin red lines that he was only able to move about 5 or 10% of the way towards the nearest target on his own. So almost all of this movement is coming through the brain-controlled stimulator. So this was 6 years ago now that we published this, and I'm happy to say that the results of this are finally being used in the first human trial, and that's going on at The Ohio State University. And what they've done, again, implanted a Utah ray in the brain. That's now an FDA-approved device. And rather than using implanted stimulation as we had envisioned and had an artist do this rendering for our study, they're beginning by using surface FES stimulation, but a very complex array of stimulators, a multi-channel band of stimulators basically surrounding the entire forearm. And their goal is to restore individual digit movement under control of the brain. So it's gratifying for me to see some of this work now moving into human trial, and certainly quite hopeful that they'll be able to reanimate the grasping of the hand under brain control. So I want to tell you about the final portion of the talk, the final story that I have for you, and that is to connect this activity recorded from the brain, as we showed, not to the peripheral muscles, as was in the last example, but immediately back to the spinal cord just below the level of the injury. And there are several significant advantages to stimulating within the spinal cord as compared to stimulating in the peripheral muscles. The first advantage, as was shown by Vivian Mouchouar over a decade ago now, in her case in the lumbar spinal cord, is that stimulating within the spinal cord gives you a more natural recruitment of the muscle fibers. So we know that FES leads to rapid fatigue in most cases. It leads to difficulty in achieving a precise force. Force likes to start from very little and jump up to quite high levels. With intraspinal microstimulation, these fine wires placed within the spinal cord, you can achieve smooth grading of force and fatigue-resistant contractions. And I'll show you an example on the next slide of Vivian restoring walking to some of her animal models with very little signs of fatigue. The second thing we like about intraspinal stimulation is that it elicits functional synergies, or groups of muscles that might want to be activated together for a task. And so I'll show an example of a grasp synergy or a pinch synergy in one of our monkey models. We'll show some examples of reach synergies in our rodent models. And the thing that we like about this is, rather than needing to activate, for example, all the muscles of the arm or the leg to achieve a given movement or posture, we can activate a single site within the spinal cord, and the spinal cord circuitry will take care of activating the rest of those muscles. So in Vivian's example, she has cats walking on a treadmill, either anesthetized cats or cats with a spinal cord injury, and with only 2 stimulating electrodes in the spinal cord targeting each leg, she's able to get the cats to step alternately. And you can imagine now it requires 8 or 10 muscles in each leg to perform this movement. So rather than having to instrument each of those muscles, she's able to have a much simpler circuit, which when we think about controlling this from brain activity is an advantage for us. And so in this walking paradigm, Vivian showed nicely both the coordinated walking but also the fatigue resistance, and that's shown on this graph here. Here she has a cat who's walking in sum over a kilometer. This is the distance in meters plotted here. This is how many times it's repeated a 3.5 meter walkway, which happens to just be the length of her laboratory. And in this cat, Vivian's stimulating within the lumbar spinal cord and basically seeing no signs of fatigue in this walking. And by contrast, when she stimulates the muscles of the cat can only walk about 100 meters, which should be shown here. And the cat, by the time it reaches 100 meters, it would basically be completely fatigued. And I think that's similar to the experience that most people have seen in the clinic with FES. If you have a walker, you could use FES-assisted walking or perhaps cycling, but the endurance of the muscles, because we're activating them in an inverse order, we're activating the fatigable fibers first with muscle stimulation, we fatigue that muscle quite rapidly. So it's a very exciting promise of intraspinal stimulation to be able to activate the nervous system without fatigue. And with that promise of intraspinal stimulation, we set out first in our monkey model to look at what types of hand and arm movements we could evoke, and then later follow this up with more experiments in the rodent. And this video shows an anesthetized monkey. This monkey does not have a spinal cord injury, and we're stimulating within the cervical spinal cord, segment C7, at a single site. And what you'll notice at first are very subtle twitches of the thumb and the fingers in a grasping movement, and then we'll turn the stimulation up, let it run a little longer. That's what you'll hear on the audio. And you should see a more pronounced grasping and ending with a prolonged grasp. You might have to turn the volume down just a tiny bit. This one's kind of loud. That's a tetanic contraction. At the end, you just hear the stimulator cycling over once per second. So these mapping studies I alluded to, we performed by passing an electrode down through the spinal cord and exploring the movements evoked every 200 micrometers down each track, and we ran a total of 24 tracks. So the gray bar is where the electrode passed. The colored dot is coded over here, in this case extension of the elbow, closer to the central canal, movements of the wrist and digits shown in orange and yellow out here in the ventral horn where you'd expect the motor neurons to be. This is just one segment, one slice of the spinal cord at C7. We expand that out to a 3-dimensional map, And we see a wide array of movements from segment C3 through T1, so third cervical to first thoracic segment. And I think it's easiest to appreciate that if we look here at the individual movements broken out. So movements of the shoulder up by C3, just as you would expect, movements of the digits clustering down here in the lower cervical segments shown by red and orange. So quite a wide variety of movements that we can evoke throughout the cervical spinal cord. We were really the first group to explore this cervical intraspinal microstimulation. We can grade the movements very nicely, as we talked about before, with fatigue resistance. Here's an example of extension of the elbow, shown at all these colored dots here. And what you'll see in the video is 3 different levels of stimuli. First a light stimuli, then a medium, and then a strong stimuli. So we can very precisely grade the amount of force. Of course, many levels in between that we could use. But that's in contrast to FES, where the muscle generally appears to be on or off. The second video just shows another functional movement, which is flexion of the wrist. This particular movement was only shot at 1 intensity there. It was a critical question for us whether this variety of movements would persist after spinal cord injury, because a lot of the research had been done in uninjured animals, anesthetized animals. And so we performed that same lateralized C4 contusion injury, and then we waited different periods of time for different animals to see if this rich variety of movements shown by all the colored symbols here, again mapped in the legend, in the intact animal would persist after injury. And we were mortified to see that 3 weeks after injury, indeed it did not. There was really only 1, maybe 2 movements that could be evoked from the spinal cord. It was an extensor synergy. It was present throughout the entire spinal cord, likely due to, it's too late to be spinal shock, but likely due to the early phases of the injury. But we were quite comforted to see that by 6 and certainly by 9 weeks after injury, this variety of movements began to return, and there was no statistical difference at 6 and 9 weeks compared to the intact, other than this massive difference here at 3 weeks if we plot the number of shoulder, elbow, wrist, and digit movements that we can evoke. You can see the injury cavity here is on the dorsal surface, centered at C4, so obviously we're not evoking any movements from the injury epicenter itself, which we didn't expect to, but at least the variety of movements have come back, and that's shown here in this video. So my grad students had fun with the animation, but they're showing here again wrist flexion. This is an spinal injured animal, now chronically implanted with wires. This is a head connector to our amplifier and stimulator. We see digit extension, which again countering that typically flexed posture. Digit extension may even be more important than digit flexion. These are very subtle twitches. Apologize if you missed those there. These are just test pulses. And then we see even things like ulnar deviation and movement of the 5th digit there. So even very precise activation of abductor digiti minimi, the muscle on the side of your hand here. So quite a rich variety of movements even after injury. And so with all these pieces together, this is the last bit of data I want to show you. We've now in the lab successfully connected brain activity to trigger spinal stimulation after injury, and seen that this circuit, what we're going to call a brain-controlled spinal interface, or BCSI for short, is capable of improving an animal's ability to reach out and perform a functional task. And before we make the connection, we look for neurons that are related to, in this case we're going to show you a lever pressing task as the functional task rather than pellet retrieval. We look for individual neurons that are related to lifting the limb, for example, preparing for the reach. The red line is when the animal makes contact with the lever. Pressing the lever, this particular neuron is active during that time, and holding the lever, there's another neuron, a third neuron we can identify that's present during that time. And so the brain codes a very rich variety of information about these motor tasks, and we can use that rich information to detect activity, in this case crossing a simple threshold in one of these neurons, to trigger stimulation delivered below the level of the injury. So again, another schematic version of our brain-controlled spinal interface, activity from the brain in real time, stimulating near the motor neurons for the arm, also activating, I'm sure, some spinal cord interneurons. And so this final video will show what an animal looks like when the stimulating circuit is off. And the audio you'll hear is just the timing of the neuron, the individual action potentials of this neuron, so you can hear that it fires more activity when the animal's attempting to perform the task, while the animal's unable to perform the task. When the stimulator is turned on, you'll hear a buzzing sound. That's the actual audio code of this high-frequency 300 hertz stimulation delivered to the spinal cord, so you'll be able to hear when the stimulator is active. So first video shows the stimulator turned off, so you can get a sense of the animal's baseline function. The clicking is the neuron. So she's trying to depress this lever. This is the injured limb, unable to depress the lever without the stimulator. Now you'll hear a buzzing sound when the stimulator is active. And you'll see that she can robustly depress the lever under brain control. And we'll play this again at half speed so you can appreciate the deficit. She's unable to weight bear on this limb, unable to extend the wrist, unable to extend the arm through the arena to press the lever. You see her stumble a bit on the arm when she attempts to use the other hand. And now when we turn the stimulator on, same clip that you saw before, but slowed down, you see she actually stimulates to weight bear on the floor, as well as to press the lever. Just one final example. Good. So we're quite excited about the functional improvement when the stimulator is off, shown in blue. Animals barely able to touch or deflect the lever when the stimulating circuit is turned on, robustly able to depress the lever, as shown by the images above. And so what we're working on in the lab now is to try to make this device small and implantable. There are a lot of advantages to keeping the signals inside the body in terms of the amount of data that needs to be streamed through the skin. Obviously, for a clinical device, this would need to be fully implanted under the skin. And so we're working with some very gifted computer scientists, Josh Smith, Adrian Fairhall is a computational neuroscientist, to try to develop a tiny implantable computer, again about the size of a pacemaker, which could actually do the computation, the mathematics, to convert brain activity to spinal stimulation. And our goal is to actually keep the computation fairly simple, let the brain do the learning, let the spinal cord adapt to that stimulation, and then have a fairly low power link to the outside. So low power, in fact, that we might be able to power the entire circuit wirelessly. So we maybe actually a batteryless implant that never has to go in for a battery change, which would be great. So Josh Smith is an expert in wireless power, and then he can gather data in the backscatter of that wireless power. So we're quite excited about this project. Goal, of course, is to reanimate the limb, but we're also looking very keenly at whether we can induce plasticity in any spared pathways that might bypass the circuit, or bypass the injury. Good, so I want to summarize there, so we have plenty of time to take questions. So the three things that I told you about today, we began with a story about epidural stimulation, I think a very exciting story from Susan Harkema's lab, where epidural stimulation and then in our own lab intraspinal stimulation was able to enhance movements in people with clinically complete but likely not anatomically complete injuries. And the question is, can this type of spinal stimulation lead to long-term recovery, lead to recovery that persists beyond the injury? Then we showed some examples of brain-controlled interfaces or brain-computer interfaces where activity recorded from the brain was used to control computer cursors and robotic arms, as well as muscle stimulation in our animal models. And now we're seeing the first human trials of brain-controlled muscle stimulation. I think it's still an open question whether the control will be good enough to improve quality of life. And then we finished with a story about intraspinal stimulation to try to evoke a rich array of movements. And I'll just leave you with the question of whether brain-controlled spinal stimulation perhaps is the solution to evoking robust, fatigue-resistant movements and not having to stream information outside the body to control an external robot. And so, of course, none of this work can be done alone. There's a huge team of graduate students, undergraduates, postdocs, collaborators, colleagues, clinical and engineering colleagues. They're all listed here as a photo of the current small group of the lab here, just the core group. There's many collaborators from other departments and institutions as well. And then our funding, of course, is shown at the bottom, and none of this work would be possible without the funding. So we'll definitely thank them, and I'll thank you for your attention, and I'll be happy to take any questions. Sure. So the question is about Susan Harkema's work, and Susie said, and I would totally agree with this, that the improvements that we see when we stimulate the surface of the spinal cord or inside the spinal cord, we don't have good models of how the brain and the spinal cord work to actually explain those improvements. We don't actually know what's happening. And so it points to the fact that we obviously, of course, need more basic science, more basic understanding, but it also points to, I think, a long history of medicine where many, if not most, treatments are at least invented with very little understanding of the mechanisms, but they just happen to work. And there's, of course, courageous people who are willing to take those risks and just try things. And in Susie's case, the discovery was a complete accident. And I'd say in most science it is as well. You need people working in the field trying new things, but often you discover something that you did not intend to study, right? It happens to be this very serendipitous discovery. Because I think if we'd waited until the theories were there to say, whoa, if we stimulate it here, we could change serotonergic input or whatever. It could take another 10, 20, 100 years, right, until those studies would be done. So I don't think we need to understand it to use it, but of course it would be fantastically satisfying to understand what the mechanism is, and probably help to guide the refinement of that treatment. Sure, so the question revolves around some of the early work done by Emilio Bizzi, where he began stimulating the frog's spinal cord actually, and saw these fantastic synergies. He could get the frog to, with a single site, he'd get the frog to make jumping movements or wiping movements, which is sort of their when they wipe a fly off their forehead. And then the question that Marge asked gets to what is the specificity like within the spinal cord? And so what I can tell you is that from deep sites within the spinal cord, where the motor neurons would lie, the ventral horn, lamina 7 and 9, you get movements that look more like muscle activation, more like nerve stimulation. And from very deep sites, you get things that look like nerve stimulation. You're actually on the ventral roots themselves. And from the intermediate lamina sort of 4, 5, 6, and so, where you expect the interneurons to be coordinating local circuitry, you get more complex movements. And so that's where Vivian gets the stepping movements. That's where we get synergies. I didn't have time to describe it. In the monkey, we would often get a reach and a grasp. That was the most common synergy, extension of the arm and grasping of the digits. It was actually sometimes hard to separate those. It was sometimes hard to get just grasping or just reaching. They always seem to want to go together. And that, I think, are these spinal circuits, whether you think of them as central pattern generators that might coordinate locomotion, or perhaps there's the analogous central pattern generator in the upper extremity that coordinates feeding, reaching, grasping, retrieving items. I think that we can tap into those with intraspinal stimulation, which we may not be able to tap into them with surface stimulation, or certainly not with peripheral nerve or muscle stimulation. Sure, yeah, so lots of great questions there. The first one, to quickly summarize, is at this point we're working on a motor control signal, an output signal. What about sensory feedback? And there are people working on that. We've just started to look at that in our lab and with our group here on campus. But there are studies showing that by stimulating the brain in human subjects with electrocorticography grids as well as animal models with penetrating electrodes, that people can learn to interpret at least different sensations that come back to the brain. Those sensations aren't always natural at this point. They might feel like wind rushing down the arm or a tingling or a tickling because they don't come in through a natural skin receptor. They come in through a complex network in the brain. But there's certainly people starting to work on that because we realize that visual feedback alone of these movements is likely not sufficient to achieve very coordinated movement. You need proprioceptive feedback, touch feedback of the fingertips. Several potential ways to do that. You could stimulate the spinal cord above the lesion. If you could figure out how to do that, you could stimulate the brain in the sensory areas of the brain. So there's definitely ongoing work in that. And then the question is, how do you get the sensation? Do you wear a glove with touch sensors? Do you use cameras and other technology to try to capture where the limb is in space and encode that? And I think all those possibilities are open at this point. But it's an earlier frontier to try to restore sensation than it is to decode motor intention. So I think we don't yet know what the best option is going to be. Sure, yeah. So the question was about the collaboration with Phil Horner. So Phil Horner is a neuroscientist here on campus in the neurosurgery department. And Phil's expertise is in stem cell therapies for spinal cord injury. And so several years ago, Phil and I were funded to combine electrical stimulation of the spinal cord with stem cell therapy in the spinal cord. And the idea was that you could put the stem cells in, but without natural activity, they might never make appropriate connections. They might not even survive, right, if they're put into the injury environment or near the injury environment, if there's very little activity or abnormal activity. And so we've done several experiments where we've stimulated the spinal cord with the stem cells present. The good news is the stimulation doesn't make the stem cells die any quicker. The bad news is, to date, we haven't seen a lot of advantage of the stimulation. So the stem cells survive. They may go through a slightly different differentiation pathway than they would without the stimulation. There's early hints that they become more faded, more differentiated towards neurons, which is a good thing. But as yet, no improvements in function that we've been able to document, or total improvements in cell survival. But I think it's very early days in those experiments. I think we're some of the first people to stimulate a stem cell in the spinal cord. I think the first people. There are a group, Christine Thomas, the Miami Project, is stimulating peripheral stem cells in motor neurons and seeing very nice effects, that the peripheral stem cells will grow and survive better with stimulation. But as I'm sure most of you know, the peripheral nervous system is a slightly easier population of neurons to work with. They're a little more robust. They like to grow back. The central nervous system is a challenging place to get cells to sprout and survive. But there's a lot of work going on in that domain as well. I guess, sorry, your final question that I skipped about the interaction, electrical versus, say, neurotransmitter interaction. At the moment we're stimulating electrically. There's new work in optogenetics, which we're a part of, to optically stimulate and activate neurons to try to get them then to release their own natural neurotransmitter. And the final bit in terms of recording that relates to your question is there are electrodes that are what called neurotrophic electrodes. So they actually have neurotrophins in them, and the neurites grow up and interface with those. Hard to record individual spikes, individual action potentials with those electrodes, but you can record local field potentials for a very long period of time. And that's Phil Kennedy's work in Atlanta. Used mostly for speech prostheses at the moment. Yes, the neurotrophic electrodes last for a very long time. Unfortunately, they're also very large, so you can only get about 1 or 2 in the brain region that you're interested in, as opposed to hundreds of the pure metal electrodes. But lots of work going on trying to figure out how to make these electrodes more robust, how to prevent the brain from reacting to them and forming scar around them, which are the 2 failure mechanisms that we think are the most likely. Sure. The question is about how long does a stimulation last in these different models, and there are some limits to stimulation. Obviously, battery life and things like that in the implanted system will require battery changes and updates. There is a small sheath of scar tissue that forms around these stimulating electrodes, and that requires you to turn the current up ever so gradually. It's the same problem that deep brain stimulators have. Very gradually over time, a deep brain stimulator has to increase its current. And in some studies, you could reach a maximum, which would be an unsafe current, right, where you begin to split water after a period of a year or perhaps more. And so there needs to be some work in that, but stimulation is much less sensitive than recording to this encapsulation. So I think it's less of a challenge. Often with a stimulator, you can just increase the current slightly and achieve the same effect that you did beforehand. Great. If that's all the questions, I'll thank you again for your time. And